So this time I'm going to be talking about data analysis, which to me means the statistics that I use. Is of course going to have a particular emphasis on the statistics of high throughput mouse phenotyping. I would imagine in many cases this is applicable to a lot of the statistics you might need, but of course you do need to take every situation as an individual case and think about it and get advice as needs be. So I'm going to be covering the role of statistics, a few words about R, which is my tool of choice, and then some talks about the different statistical methods, both numerical and categorical parameters or values. So the role of statistics. What it is really is to detect, is there a difference between these two things? This is what it normally is. But you have, in the case of high throughput mouse phenotyping, you have generally some baseline wild type mice, and you have some mutant mice, and you have something that's measured in both of them. And the question is has the mutation caused an effect? The thing is, this data set is very noisy. And the noise is multifactorial. So there's lots of different types of noise, from experimental, like the machine. Is the machine different between one measurement and the next, or is the experimenter? And there's biological, of course. There's simple biological noise that no two creatures, even if they were genetically identical, would be exactly phenotypically identical. To biological noise that is introduced by the protocol, like is there a dietary change, or is the housing slightly different? So you have all these different types of noise that make it harder to detect the real phenotypic effect. The other thing statistics do is gives you some sort of measure of how confident you are about any reported effect. So this is basically the p-value in all the statistical tests I'll be talking about. And the statistical package that I use, and a lot of scientists, I'd say probably the majority of people that once they go beyond, if they've gone beyond Excel for their statistics, most people would be using R. There are other ones out there, particularly Pay4, SPSS, MATLAB, things like this, but R is very good. It's very powerful. It's open source, and it's free, and it's very well used in scientific circles, and all the methods are well verified. And importantly, particularly for me, there are very many ways to interact with it computationally. So in the first instance, for you're likely to be, and for me, and in the examples here, I'll show you how to do one analysis. The point is, with R, it's very easy to scale that up to doing the same thing 1,000,000 times and keeping the result. And I find this very, very important. To get it, just Google R. It'll be the first one that comes up. There's a couple of IDEs that you might well find useful. RStudio is the one I use, but really that's just a way of sort of collecting the various things that you use around R. Still, the way you interact it is on the command line. As I'm going to show you during this course the actual commands that you need to execute. R Commander is another level of abstraction. So it actually allows you to use all the R tools, but in a more familiar visual way, in the way you might expect with perhaps a word processor. So you can just select your sort of data, and it actually abstracts you away from the command line interface, while still actually, of course, it's actually still doing the command lines. So if you need to, you can go to the commands it's executing and manipulate it that way. And so first, we need some data. Just as an example, we'll get it from Europhenome. So here, in this instance, all I'm really interested in is the gene PTK, and I want to know about its grip strength. It's not actually annotated here at the default level, so I click here go and look at the data, and I look at this graph, and I think, oh, look at this. I think in both the genders, the grip strength is a bit reduced. I want to look at this in a bit more detail and see if I think there is a real effect. To get it, I click on the View Data button. This gets me to a tabular view. And at the top, there is a link that gets you to a comma-separated values file, which R can read in. So here, this is when we start using R. We run something like R, and we load the data in. So this is saying, create an object called PTK, and read the CSV file, which is that URL, which I've just copied and pasted from here, and that puts the data in. Just so I can show you some of the tools, some of the different analyses, I'll take just the males of these. This is both the males and females, and this makes a subset, which is just the males. The first test we'll look at is the T-test. I'm sure most, if not all of you, have heard of this. What this does, it estimates from all the data what a single normal distribution might look like that this would be derived from, and then it gives you a probability that given that this is just one distribution, as in there is no effect of the mutation, what's the probability of getting the test and the control and test populations as far different as we really see, as in the bars on that graph as different. It does assume normality, as in they're distributed, a normal distribution. This is the bell-shaped Gaussian distribution that we often see and is frequently assumed to be how most biological parameters are distributed. And to do this in R, we say do a T-test, a T is the object, and do a test. This is the standard R syntax that means analyze the value by the genotype. The genotype here would be baseline or mutant, and the value is the grip strength. And take this data. See, here I'm only using the male animals. And this is what R produces, the critical thing being a p-value. And this is quite small. This is 2 1,000ths. So that's looking like there really is quite a significant effect there. The T-test, the pros, simple to do, both computationally, it doesn't take the computer very long to do it, and it's quite easy for the humans. This is actually implemented in Excel and everywhere, and it's quite easy to do. It is well understood by the community. An awful lot, if not the majority, of results in scientific papers are likely to quote the T-test. So this means it's easy to make the results understood to your colleagues and the wider community. And if the assumptions are met, it is a very powerful test. It will be able to detect mutations as accurately as possible. The cons, if the assumptions aren't met, both independence and normality, it is susceptible to false positives, which is what I'm certainly most worried about. I really, really don't want to be saying some mutation is causing an effect where it isn't. And there's no way to incorporate more information about the subjects. You saw I only used the males to generate in that test. There was no way of incorporating both the males and females, given that they have inherently different grip strengths, into one test. Another test I might use would be the Wilcoxon Ranksum test, also called the Mann-Whitney U. Here, you basically put all the results in order, and then you see, are one group more towards the end of that list than the other? So in this case, because the mutants were lower, you'd actually see more of the mutants at the lower end than the higher end. And this actually gives a p-value for finding that difference in distribution as much if given that they were all one distribution. It doesn't assume normality, which is the critical reason I've tended to use it, but it does assume independence. When I've looked at a lot of the parameters in urophenome, I find a lot of them are not really normally distributed. So it is something worth bearing in mind. To try it in R, this is what you do. Basically, all I've changed is T at the beginning to Wilcox. Everything else is the same, and it produces a p-value. This is actually 10 times bigger than I got with the Ttest, so it's lost, but it's still quite significant. But it is worth bearing in mind that, so this gives me pause to think if I'm really doing this analysis. So the pros, again, it's very simple to do. If anything, it's less computationally intensive than the Ttest, but it's quite easy to do. It's easy to understand, whereas it's not used as much in the community as the Ttest. It's used quite a bit, and importantly, the results look very similar. So it's easy to explain what you're doing. It doesn't assume anything about the distribution. So what I find is that many of these distributions are skewed. So when you draw the bell curve of it, it's all to one side rather than in the middle. And that is something that makes the t-distribution fail. So it's the sort of thing that means you wouldn't want to use a t-distribution. And it is quite powerful. Even if the test is even the distribution is normally distributed, so the t-test was valid, you only lose about 5% of your power using the Mann-Whitney instead of the t-test. And if the distribution is even quite skewed in quite a small way, it is actually more powerful. As well as being more selective, it is more sensitive. The cons are much the same as with the t-test. It's susceptible to false positives if the assumptions are broken. Of course, we've got less assumptions because we're not assuming the normality, but still we're assuming this independence. And again, there's no way to incorporate more information about the subject. I'm still just looking at the male animals here. If I want to incorporate more data, in this case, what I'm saying is, I looked at the graph, both the males and females had lower values. I want to incorporate that into the test. But I know the males and females have different values inherently. So what I'm going to use is what's called a linear model. This models the data as the sum of a number of fixed effects, like whether it's male or female, and then each value then has an amount of normally distributed noise. So that models the general sort of biological or experimental noise that you know you're going to get. It does assume this noise is normally distributed. To try it, very similar to what I did before, except now I'm saying I want to compare the value to the genotype and the gender. However, the output is not so simple. This is a problem with this. It is more complicated. You do need to understand a bit what you're doing. This is what R produces when you type in this relatively simple string. I'll try and explain what it means. So these are the coefficients and the estimates of the coefficients. What these numbers mean, let's say it's saying for any one male mutant value, for all values, you're going to start with 68.6. Then, if it's males, you're going to add 11.9, just for being males, because they tend to, that's about the how much bigger the males are. And then, if it's a mutant, I'm going to add 13.3, which is this. Then plus the residual. That's this normal noise. This is the actual measured distribution of the residuals. Is this effect significant? All we've done so far is build a model based on the effect, based on the data. What we need to do is build a model that takes into account genotype and one that doesn't. So this line here is exactly the same as what I did before. We can forget this NA omit. That's the exact same model we built before. And then I build another one which doesn't have the genotype in it. So basically what this is saying is build two models. One, take the genotype into account. One, don't take the genotype into account. And then we do an ANOVA, which is an analysis of variance to see how similar those models are. Is the difference between these models significant? This produces a p-value, and we see here this is still quite significant. Again, it's still getting bigger the more we tune this, but it's still significant. I've used here genotype and gender. What do I need to have gender in the model? What about if there's an actual sex-dependent, a genotype-gender interaction, as in it might, for example, the mutation might only affect males. It's X-linked. So what do we do here? Just like we can assess each of these attributes and any others. It's one important one here that I could use would be weight. For example, if you can imagine these mutant animals, if we're saying they've got a weaker grip strength, is that just because they're smaller, or is it because they're the same size and have actually weaker muscles? Or are they smaller, but have even more so grip strength, even given they're smaller, their muscles are still weak? So another attribute we could put in here would be weight. Couldn't easily do it with this data set, or I didn't show you, but this is the sort of thing, the sort of question that this method allows you to answer. And in each case, you assess whether that variable should be in the model. You could, of course, just add everything. The point is that the more variables you have in the model that you don't need, the test gets weaker. But if you exclude variables that you should have, the test also gets weaker. So the method is you test each variable individually. So here we build two more models. One including a genotype-gender interaction, one without gender in at all. And then we compare it to that first model we built. If we look at it compared to it with interaction, the p-value is really quite high. This allows us to say that the interaction is not significant, so we don't need to have it in the model. If we don't have gender in it, the p-value is high. It's highly significant, so it's very low. So we do need gender in the model. So that means that first model was the right one in these circumstances. Then the question, it may be the right model, but is it any good? There's a couple of ways, many, many ways to tell that. This is the standard way. This is the output we got that I first showed you. And if we look at this value here, multiple r squared, what this number means, that is 12%. That means the model, the attributes we're putting into the model, are explaining 12% of the variation we're seeing, which is not very much. So that's not a very good sign. Another measure, as I was saying, these residuals, these should be normally distributed. If I take out the residuals out of the model and run this QQ norm, which produces a QQ plot. So this basically orders them in order of size and plots them against what they'd be if they were normal. Now, if these were normally distributed, these would be a straight line on the x equals y axis. As you can see, these aren't. So what this is saying is this really isn't a very good model. So this introduces what else could be going on. What could I have but that I'm not including in the model? And this is something we find in Europhenome is surprisingly significant. You say Europhenome, all the high throughput phenotyping data we've looked at. Here we're actually looking at a different parameter, fat mass. If we plot each of the distribution on each day that we did fat mass on one particular line over 2 years in Mnemonic, each of these bars shows the distribution. And if you look at this, they really do vary. You know, there are some that are like this. There are some that have massive distributions. There are some down here, some up here. So what we're saying here is there is batch variation. It matters which day you do it on. Why is another question. We can't answer it. We assume it's some combination of things like litter, experimenter, temperature, the conditions of the experiment. We don't know what it is, but this is the sort of problem we're approached with. The way we can deal with this, basically, what I'm going to use litter. Kind of forget what I'm doing here. This is just a way of getting the litter out because I know how Harwell named their animals. When you get the data out of mousephenotype.org, it'll have this information in it. So I'm just getting the litter out, which I'm going to use as a batch. And then I'm going to use this different function. Before I was using LM, which is linear models. This is linear mixed effect. So this is a mixed effect model. So it's averaging it's a mixture between the fixed effects, like we've had before, as in genotype and gender, with this random effect, which is litter. So we're saying we don't actually know in each case what the litter effect will be, but we think it's likely to be consistent within the litter. So basically, this is the same function as before, value by genotype and gender, with this added term, random effect is litter. Again, I do the model without the genotype in it, and compare them with ANOVA. Now we get a p-value. It is actually still just significant at the 5% significance level, but you'll notice that that's a lot less significant than all the other numbers. So what this is saying is, there might still be an effect there, but it's a lot less. Once you take into account all the things that are likely to be having an effect, it's a lot less than it looks like at first glance. If I was doing this for real, I'd say it'd need a lot more investigation before I spent any money on looking into this line. And this is the sort of analysis that I'd be expecting people to do before they invested the thousands of pounds on rederiving and re-examining a mouse line that they found within these phenotyping databases. If I do the same residual analysis that I did before, now we can see they really do look normal. So we are saying that this is probably a much more accurate representation of the data and a much more accurate estimation of the true genotypic effect than what's gone before. That's all I've got to say about numerical variables. And I'm only going to briefly talk about categorical parameters, mostly because I haven't looked at them as much. What I mean by categorical parameters, here we have touch escape. So this is when I move my hand near to the mouse, does it run away before I get to it, which most of the baselines do, or does it wait till I touch it, which most of these do? So here, there's quite a big difference. Is this difference significant? Again, I download the data. This gets it into a long list of values. Before analyzing it, I get it into a contingency table, which makes the data look like this. And there are two basic tests that are used for this to look at categorical variables currently in the phenotyping databases I've shown you. The more standard one would be the chi-squared test. This is an approximation, and it has, depending who you talk to, exactly what rule, but it's got to be somewhere between 5 and, got to expect to get between 5 and a minimum of 5 and 10 in each of these cells, which is quite a lot, and with the data sizes that we tend to have, which is mutants between sort of 7 and 14 mutants, it doesn't tend to be matched. So I would recommend never to use this. I would always use the Fisher's exact test. It's much more computationally intensive, but it's no harder for the human. And if you use R at least, that'll actually look at the data, and if it's going to take ages and the assumptions are met, it'll just do the chi-squared test. And it's actually much more accurate. In these cases where there are small expected values, it's actually much more accurate. Here, in this case, you can see this is basically 10 to the minus 7, where chi-squared, it said 10 to the minus 10. Don't actually know what the answer is. It's obviously a mutation anyway, but the point is here, the Fisher's exact test is more accurate. And that's it.